Buongiorno, grazie mille a Ossa per avermi invitato ed avermi dato questa opportunità per parlare del nostro lavoro. Allora, I'm sorry, but I prefer speaking English about my work. So I'm working for European Commission Joint Center in Ispra in Italy. And my talk will be focused on how to replace non-human primates in neuroscience, and with a, first of all, focus on one approach, which will be how to use human pluripotent stem cells, derived neuronal models, with a possibility to replace non-human primates. So I'm very happy that my previous speakers already talked about neuroscience, so it's a nice link to my talk. So I'm working for the unit of chemical safety and alternative methods, but within our unit there is also European Center for Validation of Alternative Methods, ECVAM, and I'm sure you know this organization. So ECVAM was established in 2010 based on the Directive 63, protection of animals and use for scientific purposes. So our focus is very much anchored the 3 R's, so replacement, reduction and refinement of the use of the animals for toxicity testing of chemicals, drugs, and also in biomedical sciences. In my talk, first of all, I will start from stating where non-human primates are used, how human pluripotent stem cells could contribute to the replacement of such studies, and then I will mention also a new concept, adverse outcome pathway, how we can apply this concept in the philosophy of replacement of animals, and then how to integrate all available tools to bring the strategy towards replacement of animal testing. So it was already mentioned by previous speakers that current use of non-human primates in neuroscience is first of all in the area of higher level cognitive and behavioral studies, referring to memory, attention, decision-making, reward, emotions, visual and authority perception, and motor control. And the other use is to model diseases where this dysfunction takes place. Why? Because higher cognitive function are controlled by the prefrontal cortex, which is much less developed in rodents than in primates. That is the official statement why still non-human primates are used. However, there are new tools, new possibilities, which I will try to explore how this could contribute to the replacement of NHP animals. So I am sure you are familiar with a recent transformation also going away from animal testing towards more mechanistically based human models, and this document published in 2007, it's very much advising to use human models for human toxicity, and obviously nobody has a doubt that human models are the most relevant. And the other important issue is to understand what we are observing. So what are the mechanistic knowledge behind of the apical symptoms which are observed? And I will try to explain this a little bit further. So in our lab, we are using now only human models which are derived from human-induced pluripotent stem cells. So I'm sure you are familiar with stem cells. Just very briefly, they can be delivered from fibroblasts, taking just directly from the skin. And then we have a very robust protocol to induce pluripotency of the cells. So they are just putting back in the differentiation to the state of pluripotency What it means that we can differentiate these cells in all types of organs, so liver, kidney, and brain. And the protocol for the brain is one of the most advanced. The other cells, which are very well characterized, derived from these stem cells, are cardiomyocytes. So we really think that this is a new approach with human model which can be used for patient-specific cell therapy, chemical screening, and also human disease modeling. So I'm not going through this protocol because it's not very, it's quite technical and complex, but what is the most important is that at the end we can obtain a mixed population of the neurons and glial cells, and then we characterize these cells, how do they behave more functionally, whether they are comparable to in vivo neurons. And here we have a large range of different markers which are specific for neurons and glial cells. And we know that these neurons are the same which we observed in vivo, because not only the morphology, but also we can measure function of these neurons. And here you have, there is not pointer, but maybe I can just point out like this. Here we have H9 cell line, which comes from the embryonic stem cells. Because maybe you know that before pluripotent stem cells, there were very high-level studies on embryonic stem cells. But obviously, from the ethical point of view, we are not very enthusiastic to use these cells. That's why we compare our cells, which is IMR90, again, these embryonic stem cells, and you can see that this comparison is really at the same level. So what it means that we do not need embryonic stem cells, because these cells derived from pluripotent stem cells behave in a similar manner. So what is important here, that we have different neuronal subtypes. For instance, GABA is a marker of inhibitory cells. Then TH is a thyroxine hydroxylase, which is a marker of dopaminergic cells. GLU is a marker of glutaminergic neurons, and I can go so on and so on. We have all cell types as in vivo. And besides these morphological characteristics, we also look at the neuronal function. So you have here electrical activity of these cells, which have a spontaneous activity. The only struggle we have at the moment is that these cells are not terminally differentiated. So they represent a more fetus state. And everybody is working very hard to push the cells further for the final differentiation. Another step forward is how we culture the cells. So there are different possibilities to use scaffolds, to use different supports to create 3D structure. And here is the work actually of Primates, who was my postdoc, and then he moved to Thomas Hartung lab. And we have in our lab established these neurospheres, which are here very much developed by Primates in Baltimore. And here the cells cultured in spheres are under the movement. They are in constant shaking. And this can also mimic sort of shear stress flow of the blood, and the cells are reaching a higher level of differentiation. And for instance, we struggle with having myelin formation in 2D, which here is very nice presented. You see, these are the sheets of myelin created along the axon, which is very much next step to be closer to in vivo situation. So these neurospheres are presenting the much higher level of cell differentiation. They also express synapses. Here you have presynaptic and postsynaptic part of synapse. So we know now that it's very much important to culture the cells in 3D structure. Another struggle which we had, the presence of microglia. Microglia in the brain is very important cells which are responsible for neuroinflammation. And here I was very happy to see this paper just published in Nature, where indeed there is a new protocol to obtain microglia. It is the last type of glial cells we needed to complete the whole set of the cells. And these cells behave as in vivo. They produce reactive oxygen species under stimulation. They respond to the stimulations to inflammation. So that was the last cell type missing for the culture. Another very nice example is the paper by Lancaster, where they managed to create brain organoids. It is reaching the next level of complexity because, as you can see here, neurospheres are just a mass of cells. Here, the 3D architecture of the cells is preserved. So you can see the layers of the cells here arranged as in the brain. And here you have the 3D structure, which resembles very much cortex in the brain. So that's an absolutely stunning achievement, because not only that we have all cell types, but they're also arranged in layers as in vivo. And obviously there is lots of stuff on human on the chip and brain on the chip, where we can connect different brain structure or even different organs together with microfluidic systems. So there's lots of work going on, and we hope that very soon we will be able to reconstruct also connections between different structures of the brain, mimicking the in vivo connections. There is also a very nice in vitro model of blood-brain barrier, because if we are talking about neurotoxicity and drug testing, we have to be sure the drug is reaching the brain. So here is a very nice model, and it's already based entirely on human cells, and we can first check whether the drug indeed goes through the blood-brain barrier and at what concentration is passing on the other side. So summing up, these cells we can culture in 2 and 3D models. Human-induced pluripotent cells cultured towards the organoids can recapitulate this 3D cytoarchitecture, which is very much important because it resembles in vivo situation. And I gave you already examples how neurospheres and organoids are functional because they are expressing high level of connectivity, synaptogenesis, myelinization. So this interaction and communication between neurons and glia is taking place. So what are the advantages of using such system? So first of all, I don't have time, but I would like to very much mention that these cells can be derived from the patients which are, for instance, having Parkinson or Alzheimer's disease, and we can culture their own cells, and the whole therapy and drug screenings can be very much personalized, designed for the specific patients. So these cells, they are very much very important for several strategies to overcome this embryonic nature which I mentioned, because they are not completely mature. That's one problem. And the other problem is that these cells, to be able to use them for Parkinson or Alzheimer's disease, we have to deal with the aging cells, which is another issue. However, there are some factors which speed up the sort of aging of the cells, and there are some possibilities to use them for modeling of these diseases. So obviously they can be used for cell therapy and drug discovery, and obviously they are the most relevant human models for evaluation of neurotoxicity. And another issue which I would like to very much emphasize is the standardization of these models, because this is a huge interest now in the whole world, and everybody is using different protocols for differentiation, different markers of maturation, and now together with CAT Europe and US, we had already 2 workshops which are completely devoted, focused on on the rules, principles of good cell culture practice, trimmed design for standardization of STELSEMS-derived models, and I think it's a very important issue. So here I would like to go to another very brief topic, which I am very much involved now. It's the adverse outcome pathway to understand what we are observing in vitro conditions. So this concept is a very recent one, and it describes a sequence of key events starting from a molecular initiating event, where we have to identify, based on the existing literature, what does this interaction of the chemical of drug, initial interaction, what kind of key events is triggered at the organ or cellular or tissue level, and then organ and also organisms leading to adverse outcome. What it means that based on the existing information, we try to figure out what pathways of toxicity are behind adverse outcome. Adverse outcome, for instance, can be Parkinson's disease or learning and memory deficit of children or any toxicity. Because as you know, in the traditional toxicology, the symptoms, apical endpoint is observed, and that was enough. Nobody really cared how it's caused, what is the mechanisms behind. So here I would like to give you an example. I was very much involved in developing of this AOP by EFSA, where we found the pathways which trigger the adverse outcome defined here as, this is another AOP, Parkinson's disease, I will go here, where we define this adverse outcome as Parkinson motor deficit. So there was about 60,000 of papers to review. There was a systematic call, so we had help to do it. But at the end, we were able to establish the sequence of key events starting from inhibition of complex 1 of mitochondrial chain, which triggers inhibition of function of the complex 1. And then I don't want to go into the details, but we have these key events in a linear manner which result in a Parkinson motor deficit. What it means, at the moment, the non-human primates are used for motor deficit control experiments. So we think that by having a good, reliable in vitro assay using human models, we can study these key events which lead to this adverse outcome. So we don't need to study the adverse outcome, motor deficit in monkeys. We think that if we will find the conditions where all these key events are triggered, so we are sure at the mechanistic level that these key events, if they happen, they must lead to the Parkinson motor deficit. Here is very important that we also found causative link between key event up and down. So we have a experimental, empirical data to be sure that indeed at certain conditions, for instance, mitochondrial dysfunction will lead to the impairment of proteostasis and cell death of dopaminergic neurons. And I think this is a very powerful tool. Here is another example of our AOP developed for impairment of learning and memory in children. So again, based on the literature, we were able to find the cascades of key events leading through the inhibition, which is triggered by inhibition of an MDA receptor, through these all key events until the adverse outcome pathway. And all these assays we have very nicely established already in vitro. So again, if we find a chemical which in human models all these key events are triggered, we don't need to study learning and memory, which at the moment are used for monkey are used for it. We are sure that if this cascade of key event happens, then for sure the adverse outcome impairment of learning and memory will take place. And perhaps in this way we can also, if not replace, at least reduce the use of non-human primates. So what we are doing now, we are creating the network of these AOPs, and we try to identify these key events which are common for many AOPs, because these are the most fundamental key events we would like to create the battery on. So here we have this in yellow key events which are, for instance, in this case, in 7 AOPs. And then we go into the lab and we have all these well-established assays, and then we will use this battery of tests for screening the chemicals, the drugs, which could have a potential to impair cognitive function in children, which is now entirely done by in vivo approach. So the last slide, what is the most important is actually to integrate all these new tools all together to create a powerful battery of tests to replace non-human primates and also other animals. So first of all, it's great that we have a human models for human drug and toxicity testing and modeling of diseases. We can use these models in a variety of different configurations, 3D, 2D, human microfluidics. We have lots of knowledge on the chemical or drugs through the QSARs and read across, and there are all other in silico modeling. Here is a very important block of this AOP-driven battery of in vitro tests, because here we understand what are the mechanisms leading to the disease or adverse outcome. And then for risk assessment, I'm working for European Commission, so we are very much regulatory, toxicology orientated. For us, it's also important to be sure that whatever we test is relevant to human exposure. And there are some in silico modeling which help us to do it. So integration of all this data is a powerful approach which helps us to understand of brain function. We can use this for disease modeling, and we can use for the prediction of safety of drugs and chemicals. And the conclusions. So first of all, I'm happy that already my previous speaker characterized very much the use of non-invasive methods, which we should start from. If we can have a direct studies in non-invasive method on humans, we should use this this approach as priority. We have these human models now very much advanced, which can be used for different kind of application. And we are now understanding the pathways of toxicity by applying this AOP concept, which is also a very powerful tool. So our tool, our approach at the European Commission is based drugs and chemical testing entirely on in vivo approach by using this battery of tests anchored to the key events identified in different AOPs. And the most important is intelligent integration of this new tool, which is quite powerful and hopefully soon will replace animal testing. Thank you.